Hey, Keith.
Hey Kim, how are you?
Good. So I think we're halfway through the class, and now is about time where we're going to transition from sort of the physical understanding of what's going on with ultrasound and move into the biophysical understanding of how ultrasound is acting on the brain.
Yeah, and I want the students to know as a preface, this is one of those topics where we're still learning in real time. But when Kim and I talk to people about ultrasound neuromodulation, it's usually, one of the most important questions they ask is like, how does a neuron respond to ultrasound? So I'm really glad we're doing this lecture. Should I
Yeah,
just
me
jump
too.
in?
Yeah, go right ahead. Yeah.
Okay, so anyway, mechanosensory biology. I put this little picture of a hand here to sort of indicate the first thing we think about is sort of our sense of touch. But somehow ultrasound is able to engage some of these very same systems to modulate neurons that aren't in the hand, but potentially in the brain or elsewhere. So all right, so yeah, let's ask the first question here. Like, why is it that organisms even need mechanosensation in the first place? I mean, it seems kind of obvious, but there are some things that aren't quite so obvious. So, as I mentioned, touch, the mechanical force interacting between an environment and an organism. So, like I said, you have your hand and you might touch a desk and notice that the desk is there because we have these little mechanosensory systems built into our skin and our muscle tissue. And then there's also this idea of proprioception, which really just is a sensing of self, like has my arm extended? Am I moving in space? Is my knee bending? Things like that. And of course, hearing is a popular one to think about. Kim's talked about this a bunch in the past. Here you can see these sort of large cilia structures, and each color-coded one responds to a different frequency. And these are in the ears, and they'll oscillate when sound pressure comes into them. So these are other mechanosensors. Pain is very similar to touch. They're sort of the same basic mechanisms, but the nerves that respond to pain are probably less sensitive to mechanical force and require a lot more, to the point where the mechanical force becomes noxious or sort of dangerous. And that's when these stimuli are sensed. We also have blood pressure. So even though you don't think about it, your blood pressure is actually being sensed, like your arterial and venous walls, how much blood is sort of pushing against those walls. And that can actually change blood pressure itself. So there are sort of these automatic feedback loops. And osmotic pressure is probably one of the most important ones when we think about the brain. And osmotic pressure is really just sensing sort of salt concentrations wanting or driving fluid across permeable membranes. So you can think if there's like high salt on one side of a membrane, water really wants to travel towards that salt to balance that concentration. And that's sort of how I think about osmotic pressure. And then the last one that, is something that I think people will take advantage of in the future is interoception. One sample of that is like sensing gut stretch or, physical perturbations of digestive tracts and systems. Kim, which one of these, I might have already said it, but which one do you think of most with the brain and why the brain might have mechanosensation?
Well, you talked about osmotic pressure, but I would expect that blood pressure would also be really important.
Yeah, I was thinking both of those as well. So
Mhm.
osmotic pressure, because the brain it has these fluid systems that are changing in salt concentrations. Even as we are awake, we get higher concentrations of metabolic stuff in the brain, and that has to be sensed so that the brain can clear it. And
Yeah,
then blood
Keith,
pressure.
are the mechanisms for all of these different or the same? I guess one of the questions that comes to mind is you're showing the worms there, so the C. elegans, for example, and is the way that they sense mechanical stimuli the same as for neurons, or is it more like how fingers sense
Yeah,
mechanosensation?
that's a really great question. So the cells that harbor these mechanosensors, which we'll get into in a moment what those are, they all look different. So they may be exposed to different mechanical environments. Like the cilia are a good example. we don't have cilia at the tips of our fingers, and our fingers are not really sensitive to sound pressure. And like you mentioned, for the worm, the cells that harbor these sensors are probably very different than sensors in our gut that sense mechanical tension when we eat food. Yeah, so it's a really good point. They probably all look very different. So Kim, you might be asking though also, like, what is different about these cells besides morphology? Or maybe not. Maybe I'm just
I don't know. Was I asking that? Is that an interesting question? Tell us the answer.
I was almost going to bring it up as if it were the next slide, but it's not. Let's before we get there, let's review, because this is important. What is a membrane, and like what does it look like? This is going to come up in just a moment. So I just want to remind you from our neurophysiology lecture that a membrane is made of these sort of complex molecules that are a combination of these little lipid chains these little squiggly guys here, and these phosphate heads. So these structures have both hydrophilic, so these tail sections, or sorry, these head sections are polar and actually want to be interfaced with water, so they're hydrophilic. Whereas these sort of carbon lipid chains are hydrophobic. So these really try to avoid water. And because of this dual nature, they'll actually self-organize, where the hydrophobic components will come together, the hydrophilic components will go towards water. And this actually, creates this sort of self-forming system. And that's what membranes are. They're sort of these fluid, oily structures that interface with water on the inside and the outside. And this is important because protein channels that embed within these membranes have both hydrophilic and hydrophobic structural properties. So the hydrophobic property or structural elements will interface with our sort of lipid structures here, and the hydrophilic internals to these channels may allow water or ions to flow on the inside, or charged molecules to flow across them. So just a reminder of what that looks like, and that'll come up in a moment. And a reminder that all of these channels, there are so many different channels we talked about. There are voltage gated channels, there are leak channels. Some of them conduct sodium, some potassium. Opening up each one of these will do something different. So we mentioned earlier, if you leak potassium across passively, that will lower the resting membrane potential of a cell and make it less likely to fire. But if my channel conducts sodium, for instance, that will bring positive ions into the cell and actually raise the membrane voltage. And then you may breach threshold and have an action potential. So just to remind you of kind of the basics of membranes and how ion channels act in there. Kim, maybe that's why I was bringing it up. There are different ion channels in every cell type, and this sort of membrane potential has changed in all kinds of crazy ways. Important to remember.
Okay, good.
Okay. So how is mechanical force actually sensed? So all ion channels have a varying, either indirect or sort of direct sensitivity to mechanical force. And that is probably introduced through the cell membrane. But how this happens, like it is very unclear. And I should remind you all that like Kim and I are not by any means experts in this. We have a working knowledge, and that's what we're trying to transfer to the students. But a lot of the stuff I'm going to talk about is like hotly debated, and it's more supported by scientific evidence, but it's not known for sure. Okay, so here are like some really upfront sort of basic models of how this might work. Again, here's our lipid membrane, and here's an ion channel, and in this diagram you can see an ion here. And one popular way of thinking is that there's some sort of force created directly from the lipid membrane that is transferred to the channel by contact. And that if you have sort of a pulling force in this direction on these channels, that the channel will actually open up and allow an ion to sort of flow across. And then there's another way of thinking that, what if this membrane actually bends? Does that create an opening on one side and then allow the ions to flow across? So this is bending force model. And another model that, I don't know if it's gaining popularity, but it certainly has a lot of interesting evidence supporting it, is that there are tethers between the cytoskeleton of a cell, which would be somewhere up here, and the actual sort of intracellular or extracellular portions of an ion channel. And so if there are these spring-like tethers, anytime the cell is disturbed or moves in space, these springs will actually pull on the channel and change its conformation. And you can imagine if this pulled, you might get some ion flow across here. And that's, shown actually on the right-hand side here. So I'm just going to talk about both of these to start, and then we'll talk about one more model. And this model has, a decent amount of support through simulation and experimental evidence. So I'll start by talking about it up front. And let me also mention really quick, so I'll just draw this. Like, if you imagine that curvature is important for opening ion channels, because someone will tell you this at some point, you have to think about whether or not an ion channel would even notice curvature on a whole cell, because this diagram is not really realistic, because if we like propagate this out, that would mean that the cell was extremely tiny compared to the sort of length of the membrane shown here, which isn't true. In reality, any flex on the membrane might look like an ocean compared to like a small animal. So like this channel wouldn't necessarily notice it. So you have to keep sort of an important watch on whether they're talking about global changes in a cell's sort of flex or local. Because if this were local and just a bunch of small oscillations, then you can imagine that that might be true. Okay. But anyway, let's focus on this first sort of model here. Okay, so membranes, if they were pressed upon, you might imagine that the thickness of a membrane may change. So like if I push really hard on a cell and the cell sort of compresses out, the membrane has to necessarily become thinner, right? So if there are hydrophobic parts of the molecule that are interfaced to our lipid chains here, and these sort of want to be near each other. If you look at the right-hand side here, now you have a compression of the membrane, you have a membrane thinning out in response to this perturbation, and the channel is going to want to match the hydrophobic component to this lipid chain portion. And in that case, the entire channel would have to flex to sort of meet this demand. And this idea is called the hydrophobic mismatch. So that just means when the hydrophobic component is no longer matched to the membrane itself in terms of diameter, the channel will try to meet that by squishing down. And that would necessarily change the conformation and potentially open the channel here to allow conductance. There's another sort of way of thinking about the membrane, sensing force and transmitting that directly onto the channel. And that's just sort of generally referred to as like membrane strain transduction. And the idea here is that there is an attraction or repulsion of molecules to one another, including the channel itself. And at rest, this sort of attraction between molecules is really high, right between the phosphate head group and the lipid tails here. And the idea here is that the perturbation of these is sort of nonlinear. For instance, at this point in the channel, you're getting sort of a lot of lateral pressure that attracts between these, and at other parts, it looks different. So if you get unequal attraction changes between these, you're going to pull on this channel differently. And similarly to our last model, that'll cause sort of opening of the channel. So this one's a little confusing, but just to let you know that, it's a way that people are thinking about how these channels open.
Can you just go back to that one more time?
Sure.
You have a superimposed your drawing where you're showing at some position in the vertical direction that the forces are, more negative the pressures are more negative or more positive. Yeah, so where are those coming from?
So these different attractions are really coming from the membrane wanting to organize itself into like a hydrophobic region on the inside and a hydrophilic region here. So from what I understand, the attraction is very great at this head group, you can see there's very much lateral pressures where these little molecules are trying to come together very closely. And that's to stop the water here from coming in and making contact with these tails. And then once you're on the inside of the cell, you know, these things are all hydrophobic. They're all kind of moving around. There's really no fighting water molecules to try to come towards each other. So there's very little lateral pressure here. That's how I understand it.
Yeah, that's making more sense now. But how does, you know, ultrasound coming in and changing the pressure change things here such that the ion channels open?
Oh yeah, that's a good question. So we haven't quite gotten to why ultrasound would affect this, but you could imagine if you are pushing on these sort of head groups here, where there's already like a lot of lateral pressure, that you're going to get, I guess, sort of more tugging on the ion channel itself rather than a region where there's no attraction. Like if you disturbed the inner part of the membrane, nothing would happen to the channel. But if you push on these sort of head groups, they might like go outwards. You know, they might become thinner the same way we showed in the last model. And the tension would be distributed to this upper part of the channel. I don't know if I'm really doing a good job of describing why that would happen.
Yeah, I think that's better. Yeah, thank you.
Yeah, again, it's like, it's not super clear to me. And I think these are mostly simulation derived, but it's sort of a way of thinking of it. Yeah, I guess, and we should bring that back to here though. Like, I think it's intuitive, unless I'm wrong, to think that if you push on a sort of fluid-like membrane, that it's gonna become thin. Is that intuitive, Kim? Like, I don't know. Like, we would probably both agree that makes sense, right?
Yeah, I mean, let's think of some examples. So, like a soap bubble, as the soap bubble expands, you know that the bubble itself is getting thinner and thinner, and it just gets too fragile and blows up. So, I mean, yeah, once you change the shape, or when you change the shape and squish it down, then that's just going to change the like the surface area, and then it's just by definition has to thin because it's got to spread out in order to have a larger surface area.
Right, right. And when you start to thin it out, it's like a question of like, how does the channel interact with that membrane? And wherever there are sort of non-normal parts, you might just get this conformational change so that the channel can actually meet this membrane. And that's the way that I think about it. And the soap example is a good one for that. Okay, so this one, it's like way more intuitive. It really, doesn't take too much sort of mind bending to get to. And the idea is just that channels have these long sort of repeating sequences that create filaments. And these are bound to either sort of actin or microtubule segments that are within the cell. And so, I'm just going to draw here. Kim, you can sort of tell me if this is making sense. Imagine we had these long actin filaments, and this is sort of anchored here. If you like push these, let's say you push these in this direction because ultrasound has created sort of a forward radiating force. So now these actin filaments have moved in this direction. Then this whole tether is going to pull this way. And you can imagine in this case, it might actually like close, this channel a little bit because of the way it's tethered. But if you did the opposite and you moved it this way, it might open it up. So I know these are crappy drawings, but I'm trying to give the idea that if you're tethered to the membrane, or sorry, to the cytoskeleton, if the cell moves as a whole, these channels are going to open up and close. And these are sort of diagrammed out here in different ways. I think I have one more diagram over here. So in this particular case, you can see the non-PC mechanosensory channel is bound to microtubules through this tether, and that the tether is actually directly interacting with this little gate, and that when it's pulled on, the gate actually swings open and allows ion channels to go through. So people have a lot of different ways they say this works, but it's sort of a general way of thinking. And it's called the tether spring gating model typically. And, one reason people really like it and believe it is because, so here on the left-hand side, we're actually showing you a crystal derived model of, I believe this is a trip channel. Can't remember off the top of my head, but it actually looks kind of like a spring. Like it has this sort of spring-like structure. If you can see my mouse, you can imagine sort of being able to press on this and being able to pull on it. And the pore, which is sort of shown in the middle here, so this is the pore where ion channels would flow through. You could easily imagine that if this spring sort of compressed in this direction, that this pore would open up. And it sort of makes intuitive sense that that would affect ion flow. And I think, sorry, I'm just clicking on the diagram here. Here's like a simpler cartoon that actually shows how this spring would happen for a trip channel. So here you can see if it's actually pulled on, that in fact causes this channel to conformationally change and ions can flow through. Okay, so I'm moving on to one of my, probably my more favorite ways of thinking about it that was introduced to me by Scott Hansen, and he's sort of pioneering this field. And he's a collaborator of both mine and Kim's on many different projects. But anyway, the idea here is that mechanosensors, so there are all these different channels that have been categorized as mechanosensory, which I'll tell you about in a moment. They're often not just mechanically sensitive. So yes, they sense sort of pressure, but they also usually sense chemicals, different temperatures, and even in some cases, they'll be optically sensitive. So they'll sense electromagnetic radiation. Kim, this is like a brain teaser question, but can you think of a sense, or can you think of a moment where a mechanical sense forcing channel somewhere in your body accidentally senses chemicals and like you can intuitively pick that up? It's a tough question. I don't know if you're gonna have the answer.
Oh goodness. No, I'm not thinking of it right offhand.
Okay, so I actually might be messing this up and I'm conflating temperature with mechanical force, but sometimes they're interchangeable. But have you ever like rubbed a chili pepper on like your arm or something, or gotten it in your eyes and felt like a burning sensation?
Oh, yeah, right. Exactly. Yeah.
Yeah, so I guess I messed that up because that's really a temperature sensor. And those are very typically trip channels, but those also sense mechanical force and they sense chemicals. I forget what the chili pepper chemical is. If I think about it later, I'll tell you.
Capsaicin or something.
Capsaicin. That's right.
Yeah.
That's right. So it's interesting. It's like, why would these channels be sensitive to something that artificially makes you feel like your mouth is burning and somehow also sense mechanical force? You know, it's just really bizarre. And those same channels also sense infrared light, which is really interesting. Even in the absence of heat, which infrared light necessarily generates, creating temperature. Anyway, the point is these things are really dynamic, and like, how can we think about that in terms of mechanical force?
It seems like it's going to be hard to figure out exactly how ultrasound is acting on these ion channels if they're sensitive to all these different mechanisms, especially when there's mechanical force and temperature are two candidates, if it's responding to both.
Yeah. Oh my God, is it hard? I mean, Kim, when we wrote that review recently, there was this question of like, because I argued in the paper that temperature and mechanical force, they often can go hand in hand, because when you're sort of moving material around very quickly, you create temperature rise. So Kim, like, yeah, it's going to be tough. I don't know. What are your recommendations? You already recommended last time that we need to keep parameters constant. Do you want to talk about that really quick?
Well, not really, because that could take a little while. But I mean, I think with ultrasound that, yeah, it's tricky to disambiguate those two. I just think we need to be really careful, but I don't want to digress too much here right now.
Yeah, I might be going a little long anyway. So, okay, so let me try to come up with a way to show everyone that mechanical force and chemical sensation, which can happen in one channel, might really be the same thing. I know that sounds crazy, but let's get to it. Okay, so here's from a good friend of mine, Nick Peterson, his paper in Nature Communications a few years ago with Scott, actually. So the idea was that when you're delivering mechanical force, you're delivering really two types of energy. One is that you're just adding kinetic energy to a system, kind of like hitting it with a hammer and particles start moving around, right? It's like you take a cue ball and hit a bunch of pool balls. They just scatter. So just think about that as kinetic energy. The other one is that when you push on a cell or you push on something with mechanical force, that you get this potential energy, like you get tension in the membrane, or you get tension on this spring tethered channel membrane thing that we talked about before. So these are very different ideas. And Scott and Nick wanted to sort of figure out, well, is there a way to convert kinetic energy or mixing of a membrane into channel opening? And I'm going to jump right to the point of how they discovered that you can indeed turn mixing energy into ion channel gating. And the idea that they came up with is that you have this sort of plasma membrane. So these would be our sort of phospholipid groups, or our membrane itself. And that within that membrane, you have these ordered rafts, which are typically like made of these cholesterol molecules and other molecules that just kind of want to be together. So, okay, we have these molecules that create these rafts in the membrane. They want to stay together. And within those, they're bound to these enzymes. So by having this raft bound to this enzyme, we're almost like sequestering it, because now if it's in a raft, it can't be like over here or over here. And the membrane is full of all of these little substrate molecules that, if interacting with the enzyme, would actually create a little bit of chemistry and release sort of a chemical, we'll just call it. So it'll release a chemical that might do something. So Kim, you can imagine, in class, you can imagine if I hit this membrane, that this raft, I could add kinetic energy and mix it into the rest of the membrane, and that now it can interact with this substrate and cause the release of a chemical. So that's almost like the conversion of force into a chemical release by mixing these two components together. Kim, does that make sense?
Yeah, it does, but it raises a question for me. Well, there's a couple things I want to say. So when you were describing adding the mechanical force and is adding, a little bit of energy, I mean, that sounds a lot like temperature, where with temperature, it just causes the molecules to move around a little bit more. So maybe there's a good reason why it's hard to disambiguate those.
Absolutely.
Yeah.
You just nailed it. I mean, Scott, and that's maybe like why we're having trouble. He even says, he's like, yeah, these could probably be temperature and mechanical force sensors. And you could probably imagine other modalities that might cause mixing of the membrane that we're not thinking of,
Yeah. So another option I wonder is just pressure itself, because under pressure, then, there may be, different mobility of the lipid just due to the pressure. And maybe that's just ever so slightly different, but having the same as result.
Yeah, absolutely. I mean, I agree. I think pressure could be involved. I'll throw one more into the mix here that Scott talks about a lot, is like, if you put a molecule into this membrane that necessarily interrupts this raft, and I think he'll talk about this in his lecture, but if you add like certain anesthetics to the membrane, the rafts get disrupted and then cause these interactions and these substrate releases. So in that case, you're causing chemistry to cause this chemical release, but it's still redundant with like what you mentioned as pressure and mechanical force. So these are like multimodality sensors, and that's why I really like ultrasound, working through this as a theory. And I think you and Scott are examining some of these right now, right? In a collaboration.
Mm-hmm. Yeah.
Yeah. So, I'll try to be quick about this, but just to nail down exactly what Scott and Nick were looking at. So I mentioned that these rafts, in one sense, and I don't want you to just think about this scenario, because there's probably lots of chemical reactions that are sort of bound by these rafts. But here's one example, is that you have a cholesterol raft and that you have phospholipase D. So this is an enzyme that is tethered to these rafts. And then you have a substrate. Here is denoted PC. I actually am forgetting. That's phosphatidic choline. Phosphatidylcholine is the substrate. And you have even one more molecule involved here. PIP2 is an activator of sort of the interaction between these two molecules. So it's like, what would they call that? I think they put it somewhere in here. What do you call something when it helps a reaction occur faster? A catalyst is the word I'm looking for. Okay, so you have these three molecules, they're all bound up. And when you mix them all together, it's kind of like you're adding your own mechanical catalyst and you get an interaction. between PLD and your substrate, your what did I say it was phosphatidylcholine, and then that allows for the production of phosphatidic acid. So again, force came in, mixed everything together, the interaction happened, and out comes our molecule that's interesting. And I'm going to show you papers that were published well before they came up with this theory that showed that here's a plot of normalized current through an ion channel across different voltages. So you basically have a voltage clamp and you're looking at conductance. And when you add different phosphatidic acids, so those are shown as the POPA here, you can see that the conductance across these channels changes dramatically at different voltages. So particularly with pure phosphatidic acid. So like, I mean, that's a huge amount of current shift at different voltages. So it was already known that the presence of phosphatidic acid changed the gating of an ion channel. And if that's the case, then now this channel must be mechanosensitive, because we've already shown that, you know, you can get phosphatidic acid with mechanical force.
Keith, before you move on, I'm a little confused. What's special about cholesterol? How is cholesterol different than the rest of the lipid membrane?
So Scott will talk about that in an upcoming lecture. I'd like to say that it's much more rigid and has sort of, it thickens the membrane as Scott's showing here. But I think they have properties that are very similar to the rest of the membrane. So the short answer is I don't know a great answer to that, but I do know that they have affinity for each other and they're a little more rigid.
Is it like all these lipid rafts you're referring to are made of cholesterol?
Well, in this particular scenario, they're cholesterol aggregates, but Scott has described to me that you can have different types of rafts. So we don't want to think of it as like, this is the only way that this can happen. There are probably many other molecules that have affinity for each other in a membrane. And, you know, they'll move apart when there's mechanical force and they'll come back together during rest.
Okay.
Yeah, I don't have a great knowledge of that, but we'll bring it up with Scott's lecture, I'm sure.
Okay.
Okay, so the next question is, well, how have we like examined these over time? I just want to give you guys an idea of how people are actually measuring mechanosensitivity, and what are some tools. A very popular long-standing tool for this is called patch clamp. And this is where you take a little bath and you put solution inside of it. And then on that bath, you have your neurons, or you have some sort of cell that's placed in this solution. And then you have a long glass electrode. So this is like a long glass tube that you're actually attaching directly to the membrane of your cell. And with this attachment, you can actually measure the difference in voltage between your cell, which you're connected to, or the inside of your cell, and the bath solution outside using this bath electrode. So you're basically just measuring what's the difference in voltage between the outside and inside of your cell. And once it's connected to this system, you can basically flow solution to create mechanical perturbation, or you can use little actuator systems that push on the cell. And you can ask, does the voltage or current through these channels change? And here's like a fun little diagram of what I just described. Here's sort of the glass electrode. There are many ways that you can do this. One is where you just connect and you create suction and sort of integrate directly with the membrane. So they call that sort of suction, whole cell patch clamping. And people will often say you need a really good seal, like a resistance seal between the outside and inside, so that you're continuous with the cell. And they usually say you want a gigaohm seal or better. And you can do other things like that. And just to show you a picture of what this actually looks like, here's a single neuron. I'll try to draw around it. Here's sort of a single neuron here. And here you can see your patch, your very tiny electrode actually interfacing with the cell itself. And it gets blurry over here out in space because it's a very small plane that we're actually looking at with the confocal microscope. Okay.
How do you know the patch is really, really connected?
So yeah, that's a good question. So this is the fun part where you try to get this gigaohm resistance seal. So you can actually measure the resistance between the inside of the cell and the outside, between your circuit. So you have your bath electrode over here and your intracellular sort of electrode here that is actually directly connected to the fluid of the cell. And when the resistance of that grows, you know that you're sort of connected. So you're just like monitoring that in real time. Oh, they also usually label cells with like a color. So they'll use like genetically encoded fluorophores, and they'll color their solution. So I think that helps you actually see sort of when you're touching the cell in your prep. Okay, so yeah, that's one way. You can actually measure flow through these channels. Merit Maduk is going to be the second lecture in this series, and she's going to tell you about what that looks like when we provide ultrasound. But you can actually measure current across a channel. Another way to identify, like, is a channel actually mechanosensitive, is to use some genetic perturbation and knock it out in a simple organism or an animal model. So here we're looking at C. elegans. It's like a worm model. And you might knock out a gene and then deliver some mechanical force. And then you can ask, like, does the animal notice a mechanical stimuli anymore? So in one case, like for the C. elegans, whenever you create force at its mechanosensory tip, like its nose, it'll actually turn around, like it'll about face and sort of run away. So if you knocked out a gene and it stopped doing that, then you might conclude that that gene encoded a mechanosensitive channel because the animal is no longer sensing that force. Similar methods have been done in Drosophila larvae. So here's an old Seymour Benzer studies, the sort of father of behavioral genetics, and my own scientific grandfather from who's my PI, Bill Jaws, old PI. So I like his studies a lot. He heated up this small probe and would touch it to the larvae's skin. Now I know this is temperature, but as you now know, it's very closely related to mechanical force. And when you touch the larvae, they would create this sort of bending pattern, almost like a pain response to a heated probe. And when he knocked out this one, I think it's a trip channel called painless, you could see that compared to the wild type controls, which respond in less than 1 second. So here's time to response. The wild type responds very quickly. These painless mutants didn't really notice, and they took a very long time to create this bending response. So he concluded that painless was responsive to touch. And it also turns out that painless is involved in interoception and other mechanical forces aside from temperature. Kim, you're more familiar with this, and maybe you can elaborate a little bit. But another one is just to like examine direct neural response to ultrasound itself. So ultrasound is a mechanical force. Like, what if we just try to measure the response to that? And one method that Kim has used a lot. Kim, did you actually invent this technique?
Oh, no, not at all. No, but we did look at tail movement. Is that what you're referring to, or just looking at sort of muscle action?
Yeah, would you be able to describe what's happening here on the left for us?
Yeah, I mean, Jamie Tyler did this before us. He did the cell prep work that you're about to tell us about on the right, but he also did the in vivo work. In fact, he was very gracious and invited us down to his lab and showed us how he got it all to work. But what you're seeing here is just that you're applying the ultrasound to the head of the animal and then looking for some response. And here we're looking at the tail motion. But one of the things that we've done a fair amount, and Jamie did as well, was put EMG electrodes into the forelimbs and then looked at the forelimb response. And so in many of those, the idea is that you apply the ultrasound at the motor cortex. Although, to be honest, it's not totally clear. I mean, the motor cortex is small and there's, kind of largest focal spots. This is not clear we were, isolated to the motor cortex in any sense, really, but that was the idea.
Mm-hmm. Yeah,
I have to say, some of the work that we did with the tail was just a little confusing because the tail, I think, is also responding to sort of vestibular inputs. So, it's we could maybe bring that up when we get to the confounds as well.
Sure. But nevertheless, I mean, I think you and others have been very creative in coming up with like behaviors that a mouse would do if a brain region was activated. So like they're really nice readouts, I would say.
Nice, but it's a bit
Convenient.
confusing because it's a very high level. I mean, there's a lot going on with the interaction of ultrasound with the neurons and then the circuit and then the whole animal behavior. And in my mind, we're just not ready yet for that whole animal, analysis. And we have to really kind of step back and say, hey, wait, we got to sort of debug this and figure out what's happening at the individual steps.
It's actually, I mean, it's probably perfect that you said that, and it transitions to the right-hand side. So Kim and I have moved very closely to doing direct recording from neurons. So all the problems Kim described are like less of a problem because you're actually measuring directly from where you're stimulating. And Jamie, I think, was the first person to do this. So he had an explanted brain, and he would deliver ultrasound from a transducer, and he would use confocal microscopy and a calcium dependent fluorophore. I know there's a lot going on here, but here you can see the fluorescence in these neurons after he delivers low intensity ultrasound, and they rise. So you're getting calcium influx into the cell, which, as you now know, is an indication of neural activity. And Kim, you're doing this with whole brain imaging, if I'm not mistaken.
Yeah. So hopefully we'll have a chance to talk about that at some point. So we're doing the GCamp on the intact, in vivo mouse who's awake and relatively awake. Yeah.
Yeah, and I like have really been appreciating the field moving in that direction.
Yeah, me too. I mean, it's just really confusing to me, and maybe you understand this better than I do. But when you think about the whole animal, are we stimulating neurons, or are we inhibiting neurons? Are we stimulating inhibitory neurons, inhibiting stimulatory neurons? I mean, I just can't sort that out. So that's why I'm like, well, we need to sort of debug this and go just a little bit more simply.
Yeah, like the point you're making is like the brain is a network of so many things, so it's hard to tell just looking at a tail flick what's going on with each type of neuron, right? Like it's confusing. I'm glad we're always on the same page about this stuff, moving in the same direction. Kim, let's tell people about the major channel subfamilies, just so that they have a working knowledge of like, what am I looking for when I hear about channels that triggers me to think, oh, those might be ultrasound sensitive. The very like most popular one that you hear about, they're found in animals. They're involved in all kinds of stuff. They're the transient receptor potential family. And they're called that because, they were originally discovered in the fruit fly just for being really sort of quick acting. And these are non-selective cation channels. So when they're open, anything with a positive charge, which you now know it's like calcium, potassium, sodium, all these things will flow across the membrane non-selectively. And these, again, they've been shown to be responsive to like thermal, increases in temperature, mechanical force, light, all these crazy things. And another one that's pretty popular with ultrasound are the K1 selective family. These are also often called K2P channels. And these are potassium selective leak channels. So they have a very small amount of leak. And if you add some mechanical force to them, they may increase or decrease their conductance. And you now know that that may raise the resting membrane potential and make cells more excitable. And these are also very versatile to many different types of stimuli, including chemicals and things like that. You'll probably never hear about these, but I just want you to know that they are a mechanosensitive channel. So the epithelial sodium channel and degenerin. So these are voltage insensitive sodium selective channels. So these only conduct sodium. So they're probably largely excitatory. These come up quite a bit, I think, in like tumor biology research for detecting local strain, because tumors are very hard and they start to create more space use than healthy tissue. So there's mechanosensors involved in that. And okay, this last family you'll hear about for different reasons. They're not actually found in mammals, as far as I know. So these are mostly a highly conserved channel found in single-celled organisms like bacteria or fungi or something like that. But they're really, really sensitive to mechanical force. And so people, including Zhihai Q, who we may hear about later on, they'll actually genetically encode these in other animal models to create ultrasound sensitivity. And I'll talk about that in a moment.
Keith, I just want to ask you, when you talk about these, I guess it's important to keep in mind, and correct me if I'm wrong, that it's not like all parts of the brain express all these channels. And you already just said, of course, the last family is not in humans, but it's also the different parts of the brain have different concentrations of different channels. And so that's something else that makes it really kind of confusing. I know the Allen Brain Institute has some really nice maps where they look at different animals, and you can look at the mouse brain, for example, and different parts of the brain and look at the density of different types of channels, which is really interesting, but it also sort of underscores just sort of the complexity of what's going on here.
Yeah, I'm kind of glad you mentioned that. I mean, I think some people have talked to us about looking for unbiased ways to detect how sensitive brain regions would be to ultrasound. And you mentioned the Allen Brain Atlas. Like, what if we looked at that brain atlas and looked for specific trip channels? Would that then be able to predict ultrasound sensitivity? So you're absolutely right about the diversity in the brain. It's probably why the brain responds so differently in so many different regions, because they always have a mix of these different competing channels. Okay, so just to talk about some work from Mikhail Shapiro's lab, which is really exciting stuff. I think we're going to talk about this later on. Mikhail did quite a bit of work to actually look at different trip channels and whether or not they played a role in ultrasound sensitivity. So he used CRISPR-Cas9 gene knockdown, which is really popular these days, where you use a virus to deliver this guide RNA, and it basically creates like this sort of map of the local genome where these little scissors can come in and cut stuff out and put it back in. So he basically used these viruses to knock down different channels one by one, and then he would ask, is this cell culture or these neuronal cultures, are they still sensitive to ultrasound? And he used GCamp imaging, much like ourselves and some things I've shown. And here you can see that in a control cell culture, the cells respond very nicely to our ultrasound pulse in blue, and then they come back down to rest. But when you knock out, for instance, TRPP2, you get a large reduction in this increase. And the interpretation would be that these confer ultrasound sensitivity, which is then lost when you knock it down. So that this channel is involved. And I mentioned he looked at many different channels, and he showed that they all have differing contribution to ultrasound sensitivity, with piezo 1 having the very least and TRPP2 having the very most. People have also found that trip channels in astrocytes are sensitive to ultrasound. So here I'm showing a little video, hit play here, of calcium imaging in astrocytes. And here you can see the ultrasound is off and they have some resting calcium level. And as soon as the ultrasound is turned on, they all light up and become active for a short period. And when this was quantified, you can see that in the control, so they just took these sort of non-sensitive HEC293 T cells that didn't have any endogenous ultrasound sensitivity. So here you can see across trials, no response. But when they artificially express TRPA1 in these cells, you get sort of these large responses to ultrasound. And they did also see some response to TRPC1, although it was much smaller, and no response to these other two channels. So, people are doing a nice job of screening through which trip channels are involved. And sometimes they agree, like these two groups both found that piezo1 had a very small effect. And sometimes they disagree, because Zhurhai actually also published a recent paper showing that piezo1 was involved in ultrasound sensitivity, although he may have used different pressures or different expression levels of this channel.
So Keith, when you're talking about astrocytes, we don't typically think about astrocytes as affecting neuronal signaling, except for the fact that we do, because they're the tripartite synapse. So is that sort of what's going on here, that astrocytes are affecting the synaptic signaling?
That was the conclusion of the paper. I forget exactly how this worked, but essentially, the astrocytes would activate and then release some molecule onto their neighboring neurons and increase the sensitivity of those neighboring neurons. So that if you knock down TRPA1 in just the astrocytes, then the neurons also lost their sensitivity. I think that there's been some contention about whether or not that was true, but I can't remember why. But it's interesting to note that, there are other cells in the brain that are sensitive to ultrasound that may indirectly affect neurons. So that was a really interesting study. Two pore potassium channels are the next one. So these fall into that K2P channel group I mentioned. And I just wanted to show you another example of how people have tested this with ultrasound. So here, a patch clamp was done specifically while expressing high levels of the track channel. And here you can see the current in picoamperes across the membrane, and at baseline, they all sort of look the same. And as you increase the intensity of ultrasound, measured in watts per centimeter squared, you get much larger currents across these potassium channels. And in thinking about potassium channels, typically these would actually inhibit neural activity. So maybe you would suppress activity by targeting these channels. And there's also this sort of interesting locking effect, or like basically blocking of current following stimulation. And I don't know if that's relevant, but it also appears to be dose dependent. And Jan Kubanek has also done a bunch of studies, I think, with K2P channels. So here's just an example where he looks at TREC1. I think he's also doing conductance with voltage clamp. So as he changes the voltage, he can assess how much current flows across the channel. Or maybe as he delivers ultrasound, he can measure current. And you can see that he gets much more current across TREC1, and that he doesn't get current when he puts a TREC1 blocker, so that it indeed was, current flowing across these channels. And there are many other studies. I just wanted to show you all some examples of some that are out there. The last one I did want to mention, that these large subunit conducting channels, the MSCLs, there's not a whole lot of work on them that I've seen, but Zhurhai had done some really nice work to genetically encode them in the mouse brain artificially, and he actually conferred sensitivity to ultrasound. So cells that were not normally responsive to very low levels of ultrasound could then become responsive. So he's sort of engineering the effect. And I think there's a lot of future in sort of increasing sensitivity of brain regions that are damaged or shut off. And that's sort of an exciting future that I look forward to. Okay, so with that, I will, I guess, hand it off. So that was sort of an intro lecture, setting you up for lectures by Merit Madouk of Stanford. She's one of our collaborators here, does some really excellent work on ion channel impacts of ultrasound, and also some really interesting stuff about how the membrane physically responds to ultrasound pressure that she's going to show you all. We also have Scott Hansen, another collaborator of ours, who is going to show you many different aspects of lipid mixing that we showed earlier, and how that can be impacted by ultrasound. And he's also